Okay, all right, we can start this session. Good afternoon, everyone. My name is Monica De Gaetano. I am the Irish Association of Pharmacology's president, so I'm very happy to serve for this partnership with BPS, this particular here in Belfast. So as you may have read from the announcement on the app around the website, unfortunately, due to unforeseen circumstances, Professor Patricia Maguire was no longer available to present today her talk about the use of AI and some ethical consideration. But we are very grateful to Professor Clive Page, who agreed to step in with something like 4 hours short notice. So it may not need to be presented, but in any case, for a young investigator, it's a pleasure and an honor to introduce Professor Clive Page, who is a professor of pharmacology at King's College London and the director of the Pulmonary Pharmacology Unit. Clive's main research interests are in the pharmacology of inflammation and respiratory disease. He has published over 250 papers. Clive was the co-founder and previous chairman of the board of Verona Pharma plc, an AIM-listed company developing new drugs for the treatment of respiratory diseases. He's a non-executive director of Babraham Biotechnology Ltd, as well as being a trustee of the Babraham Institute in Cambridge and a non-executive director of the board of Immunoregulation Ltd, PrEP Biopharma, and Epiendo. Clive started his early career in the pharmaceutical industry at Santos Ltd in Basel, in Switzerland, and regularly consults to both the pharmaceutical and biotech industry. Clive has previously held a non-executive board position at Cardiome in Canada and Sterling Products in Perth, Australia. Clive is a recent former chairman of the Animal Science Group of the Society of Biology and has contributed widely to the public debate about the use of animals in research. He was awarded the Society of Biology President's Medal in 2012 for an outstanding contribution to the life sciences over the previous year. He's a fellow of the Royal Society of Biology and an honorary fellow, and has been president-elect of the British Pharmacological Society between 2022 and 2024. In 2017, Clive was awarded an OBE for services to pharmacology. So today is presenting this keynote talk entitled Sir David Jack and his contribution to respiratory pharmacology, from Salbutamol to Ensifentrine. So when you're ready.
Thank you very much indeed for the kind introduction. And as Monica said, I got a call from the current president of BPS this morning having my breakfast to say, would I stand in and give a lecture at 4 hours notice? So it's a great honor for me to be here. And as those of you who've just been to the AGM, you'll have heard about Sir David Jack, and I'll come back to this, and his legacy through his estate in giving money to the society to support new PhD scholarships. But I thought it would be helpful really to understand how we got to the position that David's estate had the money to give to the BPS. And I wanted to talk to you about his contributions to pharmacology, particularly respiratory pharmacology. And I've entitled this Salbutamol, which all of you will have heard about previously, obviously as a beta-2 selective agonist, through to this most recent drug that I was involved in developing with David called Ensifentrine. So just a few conflicts of interest here, which I hope will not influence anything I'm going to say this afternoon. So for those of you who did not have the privilege to meet or work with David, here is a photograph of David. He was the research director at Glaxo for many years. And if you go back even further, David was actually the person who first started at Allen and Hambreys in the UK. And I've called him the godfather of bronchodilators because we really would not have beta-2 agonists for treating respiratory disease without David and his wider team at Allen and Hambreys and GSK. And just a very brief synopsis, of his career, David actually started off life in Glasgow. He was a technician in pharmacy. He went on to study pharmacy and pharmacology in Glasgow before doing a part-time PhD at my old university where I did my own undergraduate degree, Chelsea College in London. And he built up from being an apprentice pharmacist in Boots all the way to being the R&D director at Glaxo. He had an absolutely sterling career, and I do not mind saying here that I think David deserved a Nobel Prize because it wasn't just beta agonists that he actually developed. And if we look back at his contribution at Glaxo, particularly his contribution to medicine, it wasn't just salbutamol, the long-acting beta agonist salmeterol, the first fixed-dose combination inhaler of salmeterol and fluticasone, but also he contributed very extensively to the discovery and introduction of topical steroids, which have revolutionized the treatment of many inflammatory diseases, including in the airway. But what many people forget, he was also instrumental in the development of ranitidine, the H2 receptor antagonist, which of course was the first billion-dollar drug, and the whole series of drugs around 5-HT receptors for migraine, sumatriptan, and the chemotherapy-induced nausea treatment on ondansetron. So this is a man who I think has contributed enormously to the welfare of the planet through his, I think, focus on drug discovery and development, but also working with a wider team at Glaxo, because no individual is responsible for all of these discoveries. Now, one of the things, I apologize for the quality of this slide, but I dug this out of a letter that David sent to me when we started the discovery and development program for Ensifentrine of what the asthma treatments looked like in 1963. And you can see here that we had non-selective stimulation of beta receptors, adrenaline, isoprenaline, ephedrine. They were really primitive inhalation administration. They were often given systemically. We had very old antimuscarinic drugs such as atropine, and we had, if you consider theophylline a PD inhibitor that was given orally and intravenously, but with a narrow therapeutic window. And of course, we'd known about the anti-inflammatory effects of glucocorticosteroids since the 1940s, and really one of the major achievements, I think, for David to look back on is the discovery of topically acting steroids. Now, actually, Sir James Black, the Nobel Prize winner in pharmacology, was also another one of my mentors. He would always really challenge me and say, well, this looks very different now, or very little different in 2023 to 1963 in terms of new pharmacological entities, and compared to other therapeutic areas, actually the respiratory area has not done particularly well. We've just improved inhalation devices, the selectivity of beta agonists, muscarinic receptor antagonists, etc. So you're all familiar with this blue inhaler, which contains salbutamol. It's estimated it's used by more than 90% of the world's asthmatics, so it's had a tremendous impact on the health and welfare of people with respiratory disease. And I just wanted to remind you, and particularly the younger people in the audience, that it was actually the work with David of a lady called Valerie Cullum, who turned out and ended up as a cardiovascular pharmacologist working at Pfizer, but she was actually a placement student who first did the original experiment to show that salbutamol was indeed beta-2 selective, published in our journal, the BJP, in 1969. And I just thought I'd show you this because we use very similar approaches in the discovery and development of ensifentrine, where if you take a guinea pig, you stimulate it with a spasmogen such as 5-HT, you can measure bronchoconstriction, you put salbutamol in and show bronchodilation, you can show it's beta receptor mediated, as is shown here, and it was the first real description of a beta-2 selective agonist affecting airway smooth muscle contraction. And what David and his team did was go from isoprenaline, which was non-selective, salbutamol, which was very selective for beta-2 over beta-1, but actually what he then said, and I remember David, he gave the Lilly lecture many years ago, he said, well, the problem with salbutamol is it's short-acting. Patients have to take it 4 to 6 times a day. So working with the team at Glaxo, he put this very long chain onto salbutamol to produce the first long-acting beta agonist, or LABA, called salmeterol, which again was introduced into clinical practice more than a decade ago. And I just wanted to show you this because it's relevant to what I'm going to tell you about the assay, because we've heard a lot about reductionist biology at this and other pharmacology meetings and molecular biology. This was the assay that he used to improve salbutamol to getting to salmeterol. And this is just using a very simple preparation of a piece of isolated guinea pig trachea. You stimulate it electrically, it releases acetylcholine, it causes contraction of the tissue. And you can see here when you put salbutamol on, it immediately relaxes, but it recovers very quickly. When they added salmeterol, a long-acting beta agonist, it was slow to produce relaxation of the tissue, but it lasted 12 and even longer hours. So this was a functional assay to be able to develop a longer-acting beta agonist, and I'll come back to this shortly. The other thing, as I've mentioned, that David did was go from our understanding that cortisone extracts were anti-inflammatory in the 1940s to really developing the first marketed topical anti-inflammatory drug for the treatment of respiratory disease, namely beclometasone dipropionate. And then others in this area, notably at Astra, developed budesonide. But it was really this initial treatment or this initial introduction of beclometasone that revolutionized the ability of giving steroids topically for a range of inflammatory conditions, including respiratory disease. And I also wanted to put this up because we've had a lot of discussion over the years about how steroids work, trying to find better steroids. But the one thing that, if you go back to the original patent for another steroid from David's group, which was fluticasone, they used a technique called the McKenzie patch test to really understand how active an anti-inflammatory agent the steroid was, both in human skin, but also in experimental animals. And the idea, of course, giving it topically, was that you produced a local anti-inflammatory effect, minimized the bioavailability, and therefore reduced the unwanted systemic side effects of steroids that you would see with oral corticosteroids that have been used up until that point. And this was a very simple assay. You'd rub croton oil on the ear, you could measure skin inflammation, and if you put on a glucocorticosteroid, you could actually reduce that inflammatory response. And this was really an equivalent, and it doesn't show up particularly well, I apologize here, but the McKenzie skin blanching test, when you put a topical steroid onto human skin, you will get this intense vasoconstriction, which you see way before any genomic effects of steroids. And the reason for showing you this is that actually, if you look at this review some years ago, this vasoconstrictor activity, the potency of vasoconstrictors in human skin and indeed in animal skin, was absolutely predictive of how good these were as anti-inflammatory agents. And this is really worth reflecting on because in the entire career that I've been in pharmacology, people have been trying to better understand genomic activity, improve selectivity and safety of topical steroids. But in actual fact, what we should possibly be doing more time on is understanding non-genomic effects of steroids that are independent of any effect on translation of pro-inflammatory proteins. It's not to say they're not important, but this is actually something I think we've understudied, because without any question, the McKenzie test was predictive. And I remember David and I went back to Glaxo some years after to talk about Ensifentrine, and I'm afraid most of the pharmacologists working at that time were completely unaware of what the McKenzie test was and why it was potentially of interest in understanding and developing better anti-inflammatory steroids. So this is something I think, is really David's legacy in the respiratory field. And now we have multiple, in fact, we now have triple inhalers. We have multiple examples of fixed-dose combination inhalers of a long-acting beta agonist, such as salmeterol, more recently vilanterol, with a glucocorticosteroid such as fluticasone, which have revolutionized the treatment of patients with both asthma and COPD. And although there are other examples out there, most of these really come off the back of the studies that David's group started in terms of discovering salbutamol. So I wanted to put up, because I'm also old, I wasn't as old as David when sadly he left the planet, but David I'd gone back and known a long time, and I published this actually in the BJ Clin Farm some years ago, and I dug this out as part of the lecture I gave earlier this year, is that actually we wanted in respiratory disease not only a bronchodilator, but we also wanted something that could prevent and resolve inflammation, whether it was for asthma, COPD, and other respiratory conditions. And I put here, this is really what we needed. And I'm going to tell you for the rest of my presentation how David and I set about effectively finding this compound at the bottom, and also just to really alert particularly early career people, it takes a long time from an idea to get through to an approved medicine. So the first thing is, whatever project you're doing, you need to justify that there's a need for new therapy. And there is no question in respiratory disease, we have good bronchodilators, steroids are very effective anti-inflammatory agents, but nonetheless, they still have side effects. And particularly now there's an increasing concern about topical steroids in patients with COPD, increasing the risk of pneumonia. And of course, what we'd really like to do, and I'm afraid I haven't solved this, is that we'd like to have an oral anti-inflammatory. But really what we'd like to do is have an anti-inflammatory and a bronchodilator in a single molecule, rather than the challenges of combining two separate chemical entities in a single inhaler. Now, one way that we started, and others had tried this as well, was to target phosphodiesterases, because as we've heard from others at this meeting, there is a broad family of phosphodiesterase enzymes. We know that they can regulate both cyclic GMP and cyclic AMP inside cells. And what was really interesting is that we already knew from a variety of other pharmaceutical companies and academic research that by targeting phosphodiesterase 5, for example, you could bring about relaxation of vascular smooth muscle. We have PD3 inhibitors for cardiovascular disease. And we also knew that if you inhibited phosphodiesterase 4, for example, with roflumilast, that was a treatment that was approved more than 11 or 12 years ago for the treatment of COPD. The problem was, and it besotted the whole area, is that oral PD4 inhibitors also cause nausea and vomiting. And so this drug has never really taken off, has a very narrow therapeutic window. There is a related drug called apremilast, which was also approved for treating psoriatic arthritis. But neither roflumilast or apremilast are bronchodilator, and therefore we wanted to add something that was a bronchodilator and an anti-inflammatory in a single molecule. So again, to just some of you in the room, if you're doing, and I spoke to some placement students yesterday who had posters, the first author of the first paper of this drug was actually a placement student, Victoria Boswell-Smith, working with my friend and colleague Dom Spina, who sadly passed away far too prematurely a few years ago, and working with David Jack. And Alec Oxford, for those of you who knew Alec, who also sadly passed away recently, Alec was the chemist who'd worked with David. He won the Royal Society Medal for his synthesis of sumatriptan, but he worked with David and I initially with a company in the UK called Venalis to develop this new class of drugs for respiratory disease. And the thinking behind this, why did we choose phosphodiesterases, is that the predominant enzyme found in airway smooth muscle is phosphodiesterase 3. The predominant phosphodiesterase isoform in inflammatory cells is PD4. And if we could find a drug that inhibited both, then you had the ability to not only cause relaxation of airway muscle, to cause bronchodilation, but switch off a whole variety of inflammatory cells implicated in the treatment of respiratory disease, and therefore have an alternative to a steroid as an anti-inflammatory agent. And more recently, people have found that PD4 particularly is also present in epithelial cells lining the airway and are involved in CFTR activation and increasing mucociliary clearance. Now, the reason, as I mentioned earlier, that roflumilast and other drugs had failed was not because the concept was wrong. They failed because many of these compounds were emetic. And they were daughters and sons of the drug Rolipram, which was developed by Schering AG in Germany as an antidepressant and is still one of the most emetic compounds ever discovered. And whilst inhalation is one way of avoiding the same sort of bioavailability you get from oral administration, Nonetheless, a very large proportion of an inhaled medicine is swallowed, and if it is bioavailable and it's emetic, it will still cause nausea and vomiting. And I put this up really to show you 4 drugs that others, not us, had really taken this concept of developing a PDE3/4 inhibitor. And this drug, Zardavrin, which was discovered and developed by Beclometasone in Germany, the forerunner, in fact, to the company that developed initially roflumilast. This compound, in its phase 2 clinical trial, at the same dose that caused bronchodilation, the swallowed portion caused nausea and vomiting. And that has stopped many, many PDE4 inhibitors, even when they've been inhaled. I worked on this compound, Ensifentrine, when, as you heard from the chairperson, I was working in industry in Switzerland. Again, this was stopped for side effects. So the concept of PDE3/4 inhibition was certainly there way before David and I took on this challenge, but it was really, I think, limited because of the pharmacophore that led to the development of some of these early inhibitors. Now, David and I took a very different view, and with Dom Spina, we'd screened a lot of known phosphodiesterase in the literature, and we particularly came across this compound, Triquensin. And Triquensin was originally developed by Herxt in Germany as a cardiovascular drug. It had been into human beings and failed miserably. And that told us 2 things, that this was a drug that would not have cardiovascular liability, but nobody in this study also had any nausea or vomiting. So this seemed to us a very sensible product to try and start making analogues of in terms of developing a new compound. And the reason I showed you earlier David's slide from his Lilly lecture published in the BJCP many years ago of using this assay of contraction of airway smooth muscle, where he showed from salbutamol to salmeterol, was not only can you look at smooth muscle relaxation, you can also very easily look at duration of action. And we wanted to get a long-acting compound, and this tissue will keep actually contracting overnight if you need it to. It's a very good, robust assay. And so we use this as a primary assay for looking at analogues of Triquensin, looking at smooth muscle relaxation using the same electrical stimulated tissue. And we hear a lot at this meeting and others, and in fact the lady who was going to talk here instead of me, I'm sure would have told you, and as you heard from our current president at the AGM, AI is revolutionizing a lot of what we do, not least that you can screen thousands, if not millions of compounds. We only made 182 compounds to get to this drug that's just been approved by the FDA. So volume is not always the right approach to this. And David used to say to me that actually the enzyme's talking to you. And when you do structure-activity relationships, if you change something and the biological function changes, you need to look at that. And I once asked him, well, how do we know that this compound's actually a phosphodiesterase inhibitor? He said, I really don't care. He said, what I care about is that you're getting smooth muscle relaxation, because that's what you're going to ultimately look for in patients. But to cut a very long story short, during this 182 compounds, we came across a compound we called RPL554, and you can see here a concentration-dependent inhibition of contraction when you superfuse airway tissues, and the very, very long-lasting activity that that drug has. And I give you a typical trace here. You can see almost immediately you put the drug on the tissue, it started or stopped contracting, and that lasted for multiple hours. So we already knew we had a long-acting bronchodilator. We then took guinea pigs and we effectively put the drug in lactose powder. We measured airways resistance in vivo in guinea pigs that are anesthetized, and if you give histamine, you will increase the contraction of the airway. And you can see here, after they've had this drug by inhalation, you almost suppress the bronchoconstriction. But the other bit of the molecule, remember, is it anti-inflammatory? So we took human inflammatory cells, and we've looked at here RPL554, as it was called, compared to several other known PDE4 inhibitors, and you can see a concentration-dependent inhibition of cytokine release from human inflammatory cells. Now, one thing you need to remember here is that if you use Seguazodone, which is a highly selective PDE3 inhibitor, that drug has absolutely no effect on human inflammatory cells. The 3 is necessary for the bronchodilation, the 4 inhibition is necessary for the anti-inflammatory action. And we went on to show this. This is an in vivo model where you can sensitize animals. And by the way, not a single mouse died in the development of this drug. And we heard it yesterday about the FDA and others suggesting we actually stop using animals over time, something we'd all like to do. The problem is you've got to pick the right species, and we've relied very heavily on mice. These are guinea pigs that have been challenged with allergen, and we measure eosinophils migrating into the lung tissue, and you can see fluticasone propionate as a topical steroid really significantly abolishes the eosinophil infiltration, and that is mimicked by this compound RPL554 and is mimicked by roflumilast, the known PDE4 inhibitor that's clinically available. And we then did experiments in primates because they're naturally sensitive to Ascaris suum. And if you find a naturally sensitized animal, you challenge them with that antigen, they will then actually produce bronchoconstriction, airways inflammation, and when we gave the drug to primates, we effectively saw what we saw in guinea pigs and got good inhibition. And it was this experiment that gave us the confidence to go out and raise the money to do the regulatory toxicology and safety pharmacology to take this compound into man. The other important thing, and this paper has only just been published, and although Dom is no longer with us, he contributed enormously to this work. At the same doses that were anti-inflammatory and bronchodilator in the primates, there was absolutely no effect on blood pressure. Now, if you inject this compound intravenously, blood pressure will fall because there's PDE3 found in vascular muscle. But the drug that we developed when you inhale it is so poorly bioavailable, it's what Alec referred to as brick dust. It was not significantly entering the circulation to affect the cardiovascular system. So to cut a very long story short, we went out and raised the money to do the toxicology. And then in 2013, we published a paper in Lancet Respiratory Medicine that were first-in-man studies with the inhalation of this drug by nebulizer in patients with asthma or chronic obstructive pulmonary disease. And as I'm going to share with you now, we got it almost, in fact, from the first volunteer conducted in Holland, in Adam Cohen's group in Leiden. You can see here a profound increase in FEV1, equivalent to what you'd see with salbutamol. The patients knew instantly that they felt better, and in a placebo-controlled study, you can see that there is a very significant effect on bronchoconstriction. So we already knew, as we'd seen in the animals, that this drug was indeed bronchodilator in patients with asthma. And it's not come out very well, I'm sorry, but it's in the paper. When we took patients with COPD, we asked them to inhale this. You can see again there was a very marked improvement in lung function that was sustainable over many hours. And over here on the left, working with Professor Dave Singh in Manchester, we took healthy volunteers. We asked them to breathe in lipopolysaccharide, which was a standard early experimental medicine technique in Manchester that Dave's group developed. And after LPS, 6 to 24 hours later, there is an increase in the number of neutrophils that you can recover in sputum. And when at bronchodilator doses of this RPL554, you can see that you've got a significant anti-inflammatory effect also in humans. So the other really interesting thing, one of my PhD students, Luigi Calzetta, who works at Tor Vergata University in Rome, we did some isolated airway smooth muscle work because we knew that this drug would not be approved as an alternative to existing bronchodilators, but in the beginning at least would be add-on. This is just to show you that if you use a threshold concentration of RPL554, a threshold concentration of the muscarinic receptor antagonist glycopyrrolate, if you put them together, this is what you predict would happen, and this is actually what happens. You get a profound synergistic response. And whilst that's also seen when you give it with a beta agonist, it didn't actually, in this set of experiments, reach significance, but you can see there's still a positive interaction with the two major classes of bronchodilator. And what was particularly interesting is, and I haven't got time to show you, this is actually with large conducting airways, but we also did the same thing with precision-cut lung slices to look at small airways. We also saw a profound interaction in small airways with this drug, looking at human airway. And some of this has been published, again, to really looking at a variety of techniques to look at synergy properly. is one of my bugbears. People use this term synergy, I think, really too often without doing it properly, but we really did spend a lot of time actually doing synergy properly, and there is this profound interaction on top of existing bronchodilator classes. And again, I'm sorry this is not projected very well. I only had 4 hours to put this together, but in the phase 2 clinical trials, there's not only a dose response curve, there was a lack of tolerance when this drug was given every day for a week, but there was also evidence of this interaction with muscarinic receptor antagonists in the clinic, as we'd seen in the in vitro experiments. So that led to, in 2015, the company that we set up to develop this, and I wasn't involved after 2015, but they had to raise more than $200 million to then do the phase 3 clinical trials. And in passing, they also had to develop a more efficient formulation to use in these clinical trials. But these 2 studies, which were published at the end of 2024, looked at, as you can see here, almost 800 patients in each of these 2 enhanced studies. And these were given on top of existing classes of bronchodilator, in some cases dual bronchodilation, and in some cases, although not the majority, there were also patients taking glucocorticosteroids. And if you look at the primary endpoint, I've just shown you here the phase 2 trial, the phase 3 trial Enhanced 2, but you can see this really improved bronchodilation that was maintained across a 12-week treatment period compared to placebo on top of existing therapy. So this new class of bronchodilator was actually providing further bronchodilation on top of existing classes of drug. And the most surprising, I think, outcome maybe for many people was when you looked at the exacerbations in these patients over these 2 trials, there was actually a almost 50% reduction in exacerbations, which is not due to bronchodilation alone. And in fact, in these patients, measuring systemic markers of inflammation, despite the fact the drug doesn't really get into plasma, you get a relationship between the reduction in inflammation and the reduction in exacerbation. And I think now I know there are studies going on to further investigate the contribution of this anti-inflammatory effect on top of the bronchodilation. But remember, the drug has 2 pharmacological effects in 1 molecule. Now, if I had $1 for every person who told me, forget it, and this includes Glaxo, by the way, David and I went to see them just at the time we'd finished the toxicology, and I won't say we were thrown out of the building, but David was very upset they didn't listen to him, given what he's done in the past, because people said if you block PD3, you're going to have heart failure. If you block PD4, you're going to have nausea and vomiting. Forgetting that, as I told you, we started with a pharmacophore that was not emetic. And we also knew that the drug from toxicology was not significantly bioavailable enough to cause a cardiovascular, any change in the cardiovascular system. And that's borne out in the phase 3 trials, and indeed all the phase 2 and the early clinical trials I showed you that we started, is that there is no significant effect compared to placebo across any gastrointestinal or any cardiovascular effect. So that's not to say down the line there may be side effects we haven't come across yet, but at the moment this appears to be a very well tolerated and safe drug that is then been approved very recently in the US for the treatment of COPD. And what is very interesting is that Merck then bought Verona. Some of you may have picked this up, and just to let you know, I didn't receive anything like the amount of money that Merck paid. They paid $10 billion for this compound. Glaxo, who threw it away at the end of toxicology and said we were wasting our time, a week after the announcement that Merck had bought this drug, paid $500 million to a Chinese company that has a similar PD3 inhibitor by inhalation that's only in phase 1, with milestone payments down the line that will probably close to $12 billion. So just because they're a big company doesn't mean they always get it right. And I think this is my advice to anybody in the room thinking about trying to find their own medicine, and at the end of the day, we're all here because we want to try and make a difference, is that however many people tell you you're wasting your time, if you believe in it, you carry on. Because I can tell you, everyone's going to tell you, from investors, etc., that you're wasting your time. And there's all sorts of reasons they do that. This drug has gone really from the first paper I showed you in 2006 from an extramural year student to being approved by the FDA in 2024. So I'm very pleased to have been involved not only from David's mentorship over the years, which often included him saying, "You laddie," because if you knew him, he was a lowland Scot, and he didn't like being questioned at all, but I learned a huge amount from working with David. It was approved on the 26th of June. It's now marketed in the US, and as I say, Merck have bought the rights to now continue development of this. It had already had, earlier this year, 25,000 prescriptions, and that's rising. And there are ongoing studies with this drug in patients with asthma, with a new inhaler device, and also in bronchiectasis and CF, and I hope that we develop. But also, I highlighted the work my PhD student did of linking ensifentrine and glycopyrrolate. There is now a combination inhaler in phase 2 development for trying to, I think, use this combination, this synergistic interaction as a potential treatment for respiratory diseases where we can introduce yet another class of bronchodilator. So in conclusion, I've been around a long time. There's far too many people for me to thank, but I really want to highlight Victoria Boswell-Smith she went on to do a PhD with us, and I cannot give a lecture without really acknowledging Dom Spina because he prematurely passed away in 2016. He was really a really phenomenal pharmacologist, and he deserves all the credit for contributing to this project. My colleagues at Verona Pharma, Michael Walker, who is the Professor of Pharmacology at UBC, Louis Franciosi, and David and Alec. And in addition to, as you heard at the AGM, David's family donating the proceeds from this drug being approved and what's happened with it, they've donated a large amount of their money to the British Pharmacological Society to really support PhD students. And you heard from Mark earlier that up until now we were only supporting half of one and then it went to one. This will give us 5 PhD students a year for 10 years. Independently of this, Alec, who also has benefited his family, although he's no longer also with us, has just donated a very significant amount of money to the Royal Society, Royal Society of Chemistry, because this money's been left by his widow in order to promote that chemists actually spend time in biological labs, because this only happens with chemists talking to biologists. And my experience is that biologists know less chemistry than chemists know biology, but actually it's that interface that we need to really promote in early career. And then I'd also like to really acknowledge my big brother, Mario Cazzola, who's the head of respiratory medicine in Rome, and Luigi Calzetta for the excellent work we've done around airway smooth muscle, and the rest of my group, which numbers far more people than on here, but I didn't have time to get a photograph with them all in. So I'd really like to think that the society should acknowledge what David has done for mankind. His estate and the development of ensifentrine that I've gone through today has really allowed money to be put back into the society. And as Mark and I know, we've worked hard to get money also out of the estate that has gone to 4K universities to support different projects for early career investigators. And none of this would happen without really the vision that David had all those years ago, and not to be put off because people said, "You're crazy." So with that, Madam Chairman, I'll stop, and I hope I've kept to time. I realise we started a little late, and I'm more than happy to try and answer questions from the audience. But thank you very much for listening to me compared to who you should have been listening to at short notice, and I hope at least it's been interesting and helpful. Thank you.
So I wish to thank, on behalf of the audience, Professor Clive for this elegant and very comprehensive overview of the legacy of David Jack. Before opening the question to the audience, I have more a comment than a specific question. I was struck by the sentence you mentioned that David told to you over the years, where the enzyme talks to you and you should listen to them. That's a beautiful sentence to reflect about, because I think as scientists we should both talk to our molecules in our own field and space of research, as well as talk to each other. So you were mentioning the chemist and the biologist and clinician and so on. So I think talking is really the key.
Absolutely, and I think also David would be the very first, which is why I spent some time at the beginning about the work he did from salbutamol to salmeterol. Biology is about function, and you cannot screen compounds properly unless you're measuring things with a functional endpoint. And I think we've become so reductionist over years. We have magnificent techniques, and I'm not saying we shouldn't be using them, but that was all done. We did not know just how good a compound this was on the phosphodiesterase enzymes until much later in the program, because he said if it doesn't relax airway smooth muscle, I don't care what it does to the enzyme. And I think we have to keep that in mind as well, because we have some brilliant technology, but actually it is about the function telling you things as you change the structure of the molecule. And that is absolutely brought home in this project.
Yeah, yeah. Which is the basis of the structure activity relationship. So if anyone in the audience wants to ask a question, maybe can come closer to the microphone.
I can't actually see people anyway, so by marks.
Hi. Clive, brilliant talk. And actually, I've heard it more than once, but actually learn new things every time I hear you speak about it. I think it's a great story. I mean, really, to my mind, it underscores something we don't often mention about ourselves, that one of the key ingredients of success in science is perseverance and resilience. When all about you are telling you you're mad, don't listen if you really believe in it. And I think your story there is a really good example. And the length of time it took, lots of other mortals would have given up, but you didn't. So congratulations. And the society owes you a huge debt, because with you we secured the donation that now allows students across Great Britain and Ireland to apply for the David Jack scholarships. And as you said, there'll be 50 studentships with the AJ Clark over the next 10 years. So thank you on behalf of the BPS. Thanks very much.
Thank you, Mark. Thank you.
We have another question.
So, Clive, lovely story. Thank you very much. And how wonderful to get a drug with a new class of action on the market for respiratory disease after all these years. So you started your talk with saying that there was not too much new. Are we done now? If
no.
Not, what next?
I think I get asked this question a lot, and I have to say that particularly in COPD, what's happening now is that people are finally recognizing that you have cardiovascular comorbidities, and most inhaled drugs are designed to keep them out of blood. There is no way you can actually properly target systemic inflammation with an inhaled medicine. And the respiratory field has spent its entire life trying to keep things away from blood. And I was involved in a debate recently. If I was someone with COPD, would I go and see a cardiologist or a respiratory physician? I'd go and see a cardiologist, because they give you beta blockers, they give you drugs for your cardiovascular disease, and we now know that you can reduce steroid use, which of course we've all been trying to do for 2 decades. And now people are beginning to talk together across those 2 different areas. So I think there's still a need, and Ensifentrine is not going to be that drug, to find ways of safely targeting systemic inflammation, particularly in COPD. Now, some of the biologics that have come along are given systemically, but they're only working in a very small subgroup. So if you think about anti-IgE and anti-IL-5s, they can reduce exacerbations, but you have to have an injection. People don't really want that. They'd rather take a tablet once a day. So I think there is still more to do. But I think this at least gives us something else that you can add on existing therapy that is giving further clinical benefit. And the fact that Glaxo have now bought this PD3/4, there's another one I know about coming along. And interestingly, I think even the people who bought this recognize that if you can make this once daily rather than twice daily, that would improve adherence. So I think there is a lot more to do. I don't think this is the end of the story.
I think we have time for a last question, because we are almost overlapping with the next session. Thanks.
Hi, Clive. Really amazing talk. I'd love to hear about the history of really classical pharmacology experiments like that. You mentioned something about the biologics just there, and I was wondering whether what you think about this sort of spectrum that people are talking about, from asthma at one end and COPD at the other, and whether stratifying patients by like cellular phenotypes, and whether there's eosinophils or neutrophils infiltrating.
Yeah, I think it's a really important area. And of course, in the past, one of the biggest finds in history was when Glaxo marketed steroid beta agonist combinations in COPD before they got approval, because they assumed the inflammation here, if it works in asthma, it will work in COPD. We're now recognizing, you say there are subgroups of people who've got high eosinophil numbers. They respond well to some of the biologics that have come out of asthma directed towards the eosinophils. But a lot of these people, it's neutrophilic. The thing about this drug is it's agnostic to leukocyte type. It blocks both. But I think it's absolutely right. Not everybody's equal. And how anybody in history can believe that a patient with bronchitis and someone at the other extreme with emphysema should be put under the same label of COPD just shows you that we need to start thinking about subtypes and actually starting to target people more specifically. So I think it's an area that, as you say, the original Venn diagram, there's what some people have termed overlap syndrome. Now it's high eosinophilic Th2 type. I think is an interesting area. I think a lot of it at the moment is being pushed by marketing, if I'm really honest. I think people who've got anti-eosinophilic drugs as monoclonals in asthma are trying to see there's a bigger market out here, but there's definitely a subgroup that are responding. But I'd take you back to the original study of the monoclonal against anti-TNF. There's a subgroup that got better, there was a lot of people who got worse. And that immediately tells you that not all patients are equal with COPD. And we have to learn from that and start targeting, as much of this meeting says, around stratifying, getting the right people for the right trials, rather than accepting all comers and hoping that that's going to work.
I appreciate that. Thank you.
Okay. Unfortunately, in the interest of time, we need to wrap up this keynote session, because I know every one of us would love to hear for another hour Professor Clive Page. Please join me with a final round of applause for Professor Page.
Thank you.